Now that you know how xylem and phloem work individually, let's zoom out. Okay, and then compare how these two great transport systems work within the plant. We know that one of them carry water and ions and the other carries food. Although they sound very similar in terms of both being used for transport, the system as such is completely different. A typical plant can grow several meters tall. We have seen such huge trees that are present on Earth. And diffusion alone is too slow for anything to move within these very tall trees. So, how do plants do long distance transport of water and ions? It is through this process of mass flow. Mass flow is a process where bulk movement of fluid and dissolved substances happen, but it happens through a long column. These long columns are the cells that are part of both xylem as well as phloem. The driving force for the bulk movement of substances through the column is a pressure gradient that is created within these structures. What exactly is moving during the mass flow? So, in a potted plant, in case of xylem, there is movement of water and mineral ions that the root have absorbed from the soil. They move from the soil to the leaves. It's only upwards and the movement is unidirectional. In case of phloem, we see that there is transport of sucrose, amino acids, and other organic compounds. These are produced in the leaves, and from the leaves, they are taken to different parts of the plant, including the root. And this transport is multi-directional. Now, let's look at how the driving forces differ in both these systems. In case of xylem, it uses the nature's strongest suction pump, which is transpiration. So let's say there is a plant. Okay, so during a hot day, the excess water that is present gets evaporated from the upper surface of the leaf. The water molecules which are present within the pores of the soil are absorbed by the root. Now, as the transpiration continues, it creates the sort of vacuum or space. And in order to fill up the space, the stem pulls up the water that has already been absorbed by the root and it takes it to these different spaces in the leaves. So, as the water evaporates from the leaves, it pulls entire water column up. It is similar to drinking water from a really long straw. The minerals are also taken up through a similar route into the leaves. The water column is maintained by the cohesion between the water molecules and the adhesion between the xylem wall and the water molecules. In case of phloem, the mass transfer is a little more elaborate and there is involvement of xylem in it too. So we have the sieve tubes here. Sieve tubes also have the live companion cells. Then we have source cell, which is usually where there is a lot of food. So in this case, a leaf where the food is produced is considered as a source cell. A sink cell is where there is no food. So in this case, we could consider a root to be a sink cell. The companion cells and the source and sink cells are connected by appropriate ion transport channels. The xylem vessels hold water. First, let's see how food moves out of the source cell. We start with the movement of hydrogen between companion cells and the source cell. So, this creates a natural gradient for protons, right? Now, a proton along with the sugar is going to be co-transported into the companion cell. From the companion cell, they move into the sieve tubes by active transport. Water moves into the phloem by osmosis, and this creates an increase in hydrostatic pressure. You can say that this is the pressure created or felt in a place when water enters that particular space. And this pressure is usually very high near the source cells and low near the sink cell. The buildup of pressure pushes the sap down along the pressure gradient towards the sink of the phloem. So you can see that slowly the sugar molecules and the water are making its way towards the sink. As the sap reaches the sink, the solute molecules are moved into the sink where they are converted into other molecules like starch for storage. As the solute is removed, this makes the water to move back into the xylem through osmosis. This sort of a movement helps in maintaining the pressure between the source and the sink without bursting any of these transport tissues. The transpiration is a pulling force, whereas this sort of a mass flow is a pushing force. The flow through xylem is a passive transport, but the flow through phloem is an active transport requiring energy. Finally, the conducting tissues are also different from each other. Xylem is a dead tissue while phloem is a live tissue. In xylem, we have tracheids, which are narrow pipes that have thick walls and tapering ends. They have a wide lumen for the flow of water, and that's because they are dead cells and do not have protoplasm. They are arranged one on top of each other so that water can flow through the connecting holes in between them. Xylem also has vessels which are perforated and have open ends on both the sides. They are like uninterrupted tubes for water flow. The end walls in between them are dissolved. You can see it if you cut open the vessels. In case of phloem, sieve elements are the principal conducting cells. These are elongated cells with thin walls. Although they do have a nucleus during developmental process, they lose the nucleus as soon as they reach maturity. The sieve elements are connected end to end through a sieve plate. These plate contains pores which allows for the movement of phloem sap between the adjacent cells. Since sieve elements do not have a nucleus, the companion cells are associated with them. These are cells with nucleus that are connected to the sieve elements through simple pits, and they take care of the metabolism happening within these tissues. Here is a summary of all the differences that we have seen, and it just shows how cleverly the plants have divided their internal transportation duties. Fascinating, isn't it?